Hello friends, let us discuss vitamin K in this video. Vitamin K is one of the fat soluble vitamins. K stands for coagulation. In Danish language, which is spoken in Denmark, coagulation is written as coagulation. It begins with K, right? There are three vitamers of vitamin K. They are named as K1, K2 and K3. K1 is Phylloquinone, K2 is Menaquinone, K3 is Menadione. K1 is found in plants, that is why the name Phyllo. K2 is produced by intestinal bacteria and it is also present in food of animal origin. Whereas K3, that is Menadione, is a synthetic compound. If you look at the structure of K1, K2, K3, what are you seeing? See this. This is the quinone ring, right? So phylloquinone, mina, quinone. So there is a quinone ring. See dione. See there are two ketone group. That is why this is mina dione. Also notice the side chain. What are you noticing in the side chain? There are multiple isoprene, right? Isoprene is a 5 carbon compound. So these fat soluble vitamins A, D, E, K, they all contain isoprene side chains. Okay. All right. Let's quickly discuss the dietary source of K1, K2, K3. K3 is a synthetic compound. So leave it. What about K1 and K2? Phylloquinone, as the name suggests, it is there in plants. What kind of plants? Cruciferous and dark green leafy vegetables such as broccoli, spinach. Look at this. Cauliflower, that is gobi, cruciferous vegetable. What about K2? As I told you, it is produced by the intestinal bacteria. Okay. So our commensal bacteria, they produce K2 and it is absorbed by the human body. Also, it is present in diet of animal origin like egg, liver and dairy products. Right? Vitamin K is the only fat soluble vitamin with cofactor role. Whereas all the water soluble vitamin B complex and C, they have cofactor role. Okay. Vitamin K is the cofactor for gamma carboxylation of glutamate, right? So vitamin K helps in the production of gamma carboxy glutamate. Glue to GLA. Glue is glutamate. GLA is gamma carboxy glutamate. Okay. So hereafter whenever I say GLA, that means it is gamma carboxy glutamate. Remember, what are the proteins that contain GLA? So formation of glue to GLA is a post-translational modification. Remember, this is a PTM, post-translational modification. Lysine to hydroxylysine. That is also a post-translational modification. Which vitamin is needed for that? Tell me. It is vitamin C, right? So vitamin K is needed for gamma carboxylation of these proteins. What are they? Clotting factor 2, 7, 9, 10. How to remember them? Do you remember the cranial nerves carrying parasympathetic activity? They are what? They are 3, 7, 9 and 10. Whereas GLA containing proteins are 2, 7, 9, 10. In addition to that, protein C, protein S, protein Z, osteocalcin, periostin. So they are present in bone. Transthyretin, which is your prealbumin. I have already made a video on transthyretin. You can watch this video later. So Gamma carboxyglutamate is present not only in clotting factors but also in these many proteins. Remember this. First of all, if you look at the structure of glutamate, it already contains one gamma carboxyl group, right? Glutamate is a dicarboxylic acid. There is one alpha carboxyl group and gamma carboxyl group. In proteins, alpha carboxyl group takes part in the peptide bond formation. So it is not free. So which is free? The side chain. The side chain is free, right? So this is free. So there is already one gamma carboxyl group. Vitamin K is going to help in the formation of one more carboxyl group here, right? So now there are two gamma carboxyl group. Why do we need this? Tell me, why do we need two negative charges? So two minus charges. Can you guess? It is because calcium, which has 2 plus charge, can come and bind here. So during the process of clotting, what happens? This helps in attachment to the phospholipid layer, right? Hmm? What is calcium? Calcium is nothing but clotting factor 4, right? So it's important for blood clotting, right? In the bone also you have calcium, right? So gamma carboxyglutamate, that is GLA. Hereafter we will call this as GLA. GLA is needed for binding to calcium. That is why we are adding one more carboxyl group. Remember this important point. All right. How this addition of gamma carboxyl group takes place in the human body? So that is known as vitamin K cycle. So in the exam, they can ask you, draw the vitamin K cycle. Why it is cycle? Because we have limited quantity of vitamin K in the human body. So it is cycled, right? So cofactor, it undergoes some change during the enzymatic reaction and it is regenerated. Okay. So this is a protein that contains glutamate. Okay. So protein glutamate. This can be your prothrombin. This can be your osteocalcin, any protein. Okay. So protein contains this glutamate. You need oxygen and carbon dioxide also. Okay. So the enzyme catalyzing this reaction is vitamin K epoxidase. Vitamin K epoxidase. Remember. So from glue we get GLA. No need to go in depth how this glue is converted to GLA and all. Let's not worry about that very much. So remember, this is not a carboxylase. This is what? This is epoxidase. Okay. So in that process, vitamin K is converted to epoxide. So normally vitamin K is present in the form of hydroquinone. In the conversion of glue to GLA, vitamin K is converted to epoxide, right? So we have to regenerate this epoxide to hydroquinone. How that is done? That is done by an enzyme called epoxide reductase. It will first convert the epoxide to quinone form. Then we have another enzyme called quinone reductase, which will utilize NADPH plus H plus. NADP plus is produced. So quinone is converted to hydroquinone. So this is the vitamin K cycle. See this? Vitamin K hydroquinone is converted to epoxide, then quinone, then to hydroquinone. Right? So in this process, glue is converted to GLA. That is the significant thing. Okay, glue is converted to GLA. Right? What is the pharmacological connection between this vitamin K and warfarin? If you look at this, warfarin is an inhibitor of epoxide reductase. So warfarin will inhibit the formation of GLA, right? GLA containing proteins are also known as VKDPs, vitamin K dependent proteins. Okay, so warfarin is an anticoagulant. It's an anticoagulant. Okay, if you look at this, see this prothrombin to thrombin conversion. So this reaction is very important. It's there in the common pathway, right? So prothrombin is one of the vitamin K dependent proteins. So warfarin will increase the prothrombin time, right? Warfarin is used as rat poison. So it causes internal bleeding in rats. So the rats go to hypovolemic shock. So they will get thirsty. They will go to the water source and they will die near the water source. What about humans? Why is it used? First of all, warfarin is used in many conditions. For example, people with prosthetic valve, right, to prevent any clot. So it's used. People with stroke, right, transient ischemic attack, stroke. So all these conditions, right. So to prevent clotting, prevent the emboli, so we use warfarin. So how do you monitor the effect of warfarin? That is by periodic prothrombin time. So PTINR. What is INR? International normalized ratio. What is that? We'll discuss in a different video. What is the antidote of warfarin poisoning? Vitamin K. For acute bleeding, you give fresh frozen plasma, which will have all the clotting factors. Chronic warfarin therapy leads to osteoporosis. How? Vitamin K is needed for coagulation. How can dysfunction of vitamin K leads to osteoporosis? Can you think? You remember osteocalcin is a vitamin K dependent protein, right? So vitamin K is needed for gamma carboxylation of osteocalcin. That is why long-term warfarin therapy is associated with reduced bone mass. Okay, so this information is from Harrison. So remember this, in exam they can ask you what is the biochemical basis of osteoporosis found in patients of chronic warfarin therapy. All you have to write is this information, right? Okay. Next, we will move on to the causes of vitamin K deficiency. Poor nutrition, patients on prolonged antibiotic use. Why? Vitamin K2, that is Menaquinone, is produced by the intestinal bacteria. So, prolonged antibiotic use will affect this. Any cause of pancreatic insufficiency, that is cystic fibrosis. So, here what will happen? There will be steatorrhea, that means lipid or fat maldigestion and malabsorption, right? So, fat malabsorption will lead to fat soluble vitamin deficiency, right? Biliary obstruction, again, biliary obstruction will lead to malabsorption of lipid, so that will lead to vitamin K deficiency. Clear? Another important question for your exam is hemorrhagic disease of newborn. Remember, hemolytic disease of newborn is different, hemorrhagic disease of newborn is different. Hemorrhagic disease is vitamin K, hemolytic disease is your RH incompatibility. Okay, don't confuse these two. Menaquinone, that is vitamin K2, is synthesized by the intestinal flora. Intestine of newborn is sterile and the placental transport of vitamin K is poor. So, the neonates are deficient of vitamin K. Okay. So, when we are born, we are vitamin K deficient. Breast milk is a poor source of vitamin K. So, The newborns, especially the preterm ones, are prone for bleeding. So that is prevented by 1 milligram intramuscular injection of vitamin K at the time of birth. Okay, very, very important point. So this will also be asked in your exam. What is the biochemical basis of injection of intramuscular vitamin K for neonates? So you have to explain to prevent hemorrhagic disease of newborn. Why? Because the intestine of newborn is sterile, they are vitamin K deficient, right? I hope you will be able to write in the exam. Thank you for watching. Please share this video with your friends.